Hello students, hope you've been enjoying your snow days, and I hope that you enjoy this video about aerobic respiration and fermentation. The good thing about the video is that you can watch this multiple times to help you study, and I recommend that if you need to watch it multiple times, go ahead and do so. I will try to make it fun and try to make it educational as well. So the topics that I'm going to talk about today are aerobic respiration, picking up with the citric acid cycle and electron transport chain, and this video also at the end is going to include the information about fermentation. So here we go. So the first thing to review is to make sure that you remember the chemical equation for aerobic respiration. So take a minute to write that from memory. You remember it, right? You need to have it memorized for your exam. And if you don't have it memorized yet, here it is. So we are starting with glucose, which is a monosaccharide sugar, chemical formula C6H12O6. And this process of aerobic respiration is talking about the burning of glucose in the presence of oxygen. So we are burning glucose in the presence of oxygen for the purpose of making ATP energy for the cell. So the cell can use ATP to power many different things, many different jobs that it needs to do. And glucose is a great fuel for making ATP. And along the way, as the cell is burning glucose for energy, it's also releasing carbon dioxide and water as byproducts. So there are, for every one glucose, it takes 6 oxygen molecules of oxygen gas and produces 6 carbon dioxide molecules, 6 water molecules, and several ATP. So this is the summary of the process that represents aerobic respiration. Also recall that for both animal cells and plant cells, both of those cell types do aerobic respiration. And both of those cell types mostly complete the work of aerobic respiration in the mitochondria, which is an organelle. So here we have a cartoon of the mitochondrion, and the blue background, which is hard to see, is the cytoplasm surrounding the mitochondrion. And what I talked about in the last video was glycolysis, or sugar breaking, where we're taking the 6-carbon glucose and splitting it into 2 pyruvic acid molecules. And that process produces some ATP. And then the next 2 steps are the focus of this video today. These are the citric acid cycle. This can also be called the Krebs cycle. It can also be called tricarboxylic acid cycle, or TCA cycle. And then the final step is the electron transport chain. So these steps that I'm talking about today do happen inside the mitochondrion. So that's an important difference from glycolysis. So let's review generally what happened with glycolysis. We're starting with a 6-carbon sugar, glucose. And these balls connected by the lines represent a 6-carbon sugar, glucose. And during glycolysis, this 6-carbon sugar gets broken down into 2 separate 3-carbon molecules called pyruvic acid. So here's one pyruvic acid, here's another pyruvic acid. So glucose has been split in half, and I'm going to pull the screen up for a minute and write something here. So we have a 6-carbon glucose being broken down into 2 3-carbon pyruvic acids. And the breaking of the bonds in glucose that led to this event released some energy. So during this process, the cell had to add in 2 ATPs during the splitting process, and then it releases 2 pyruvates plus 4 total ATPs plus two of these NADH molecules that are carrying energized electrons. So the cell had to put in 2 ATP. It gets out 4 ATP, but really it has a net gain of 2 ATP because it had to put in 2 to get 4. So glucose has been split in half. 2 ATPs have been produced. 2 NADH have been produced that are carrying energized electrons. And that was basically the end of glycolysis. Then after glycolysis, there's this intermediate step. I'm going to erase this now. There's an intermediate step where each individual pyruvic acid molecule gets turned into something called acetyl-CoA. So acetyl-CoA is a 2-carbon molecule with this CoA group attached to it. And this is the molecule that's then going to go to the citric acid cycle. So before this process goes to citric acid cycle, the cell has to produce this acetyl-CoA, which is a 2-carbon molecule. So sometime between here, 3 carbons, and here, 2 carbons, there was a carbon dioxide released. So this is the first time that we're seeing carbon dioxide being released during the breakdown of a 6-carbon glucose. And also keep in mind that everything that just happened here with this pyruvic acid would also happen the same to this pyruvic acid. So there would be an acetyl-CoA produced from this pyruvic acid, and there would be a carbon dioxide released as well. Same with this one, same with this one. So that's a review of what happened up through the end of last lecture, which gets us ready to talk about the rest of aerobic respiration. And before I go on, keep in mind that we are turning chemical bond energy represented in all these covalent bonds in glucose. We're turning this source of energy into ATP energy. And it's a multi-step process, and there are many enzymes involved in this process. So by the time glycolysis and the acetyl-CoA step are finished, we have partly broken down glucose. So we are at the point where we have two carbon molecules of acetyl-CoA. And those acetyl-CoA molecules still have a lot of bond energy remaining in those. So there is a lot more energy that can be extracted from acetyl-CoA if the cell will do those extra steps. So the first part of those extra steps is the citric acid cycle. And this is my least favorite part of the whole aerobic respiration process, and I feel that it's the most difficult to understand. So let's start over here with what's coming into this process. So here we have 2-carbon molecule acetic acid. Notice the CoA has left at this point, but we still have the acetic part. This is like vinegar that you keep in your house. So we have a 2-carbon molecule coming into the citric acid cycle. And when acetic acid comes in with its 2 carbons, it's going to combine with a 4-carbon molecule. This actually is getting covalently attached to a 4-carbon molecule to make another 6-carbon molecule called citric acid. So here, this particular 6-carbon molecule is citric acid, and that's where the citric acid cycle name comes from. So it seems like, well, if the cell is trying to break down covalent bonds to produce energy, this seems like a step backward if we're now building a bigger molecule out of a smaller molecule. But the cycle part of this ensures that this 4-carbon molecule picks up acetic acid, forms citric acid, And then there are several enzymes along the way here that end up removing those 2 carbons that originally came in and regenerating the 4 carbon again. So the best way I can describe it simply is that this 4 carbon molecule holds on to the 2 carbons to allow their energy to be extracted, and in the end they are broken down and they produce these products. So for this class, you don't have to learn the irritating details of the citric acid cycle, but you do need to know that after this cycle has gone around one time, for one acetic acid that came in from glycolysis and the intermediate step, these are the results. So let's focus on the results more than the details of what just happened here. So for every acetic acid that comes in, you start with a 2 carbon molecule. You produce as waste 2 carbon dioxides, 2 CO2s. So the 2 carbons that came in, they leave as carbon dioxide waste products. The bond energies that were present in this acetic acid molecule, where does their energy go? So for this one acetic acid that came in, 1 ATP is produced, so that's good. That's what the cell wants. It wants some ATP energy. And also, for every acetic acid that comes in, we make more of these energized electron carriers. So the one so far that we've talked about is NADH, and NADH is carrying energized electrons. We also have in this step a new energized electron carrier called FADH2, and it's also carrying energized electrons. So production of these is actually the major outcome of the citric acid cycle. Yes, we have some waste CO2. Yes, we have 1 ATP per acetic acid. But we have 3 NADH and we have 1 FADH2, and these are carrying energized electrons. So I'm going to pull the screen up again and write some numbers over here to demonstrate something else. The reason I'm not leaving the screen up is that I know there's an irritating glare right here on the camera. So in this process, we are making lots of energized electron carriers, a little bit of ATP, and our waste CO2. So 2 CO2s, 1 ATP, 3 NADH, 1 FADH2. But all of these are per 1 2 carbon starting material. This amount for every 1 2 carbon starting material. So if you think about over on this side, we started with a 6 carbon glucose. We then had 2 3 carbon pyruvic acids. We then had from each of those an acetyl-CoA that was 2 carbon. Same here. And then each one of those gets converted to an acetic acid. So this output is only measuring half of what we originally started with. So we had 1 glucose, it's split into 2. Each half then ends up participating in this cycle. So let me use a different color of marker to show the totals. So in total, we're getting 4 CO2 out of the citric acid cycle, because we're starting with actually 2 carbon molecules originally coming from glucose. And we are getting 2 ATP. We are getting actually 6 NADH and actually 2 FADH2. So citric acid cycle is complex, somewhat irritating, but very productive for the cell as far as making energized electron carriers. Got 2 ATP, waste CO2, which is not really important to the cell, but we've got a lot of energized electron carriers. So, glycolysis has happened now. Citric acid cycle has happened now. And by that point, all of the original covalent bonds in glucose have been broken down. There are no more carbon chains left. All of the carbons have been released from glucose in the form of carbon dioxide, which we blow out with our breath, every breath we take. We've made a little bit of ATP, so at this point the cell has made 4 ATP, 2 from glycolysis, 2 from citric acid cycle. We have made a lot of these energized electron carriers, NADH and FADH2. There are a lot of those still hanging around, with only 4 ATP, and from the equation that you memorized for this process, we haven't seen oxygen yet, nor have we seen water yet. So, electron carriers, oxygen, water, a lot more ATP, all of that has to come from the next step in the cycle, which is the electron transport chain. So, electron transport chain. This is a useful picture to start talking about the electron transport chain, because the purpose of the electron transport chain is to get electrons from these energized electron carriers, NADH, and you could also put FADH2 here. Get these high energy electrons and use their energy to make ATP. Somehow this electron energy is going to be converted to ATP energy. And this process happens by these electrons getting passed, kind of like hot potato, passed from one protein to another in the electron transport chain, and every time they get passed, their energy is released little by little. And in the end, oxygen picks up the electrons and combines with a couple of hydrogen ions or protons and produces waste water. So we have electrons coming into the electron transport chain, they're high energy. When they leave the electron transport chain, they're low energy, and some of the energy that they lost has gone into making ATP. And your book describes this as electrons falling downhill, which is a pretty useful concept. So electron transport chain, again, we start with energized electron carriers carrying these high energy electrons. Their energy is successively decreasing as they go through electron transport chain. In the end, the electrons get attached to oxygen and we form water. And we have a lot of ATP. Okay, so let's go back and review where is this happening. So citric acid cycle happened inside the mitochondria. Glycolysis was happening in the cytoplasm, but the stuff that came out of the citric acid cycle is in the mitochondria. And fortunately enough, the electron transport chain is in the mitochondrion as well. So that's convenient for the cell. So first, I want you to look at this picture first. Don't look at this other stuff, look at this picture. This is the mitochondrion. And the mitochondrion has 2 sets of membranes. So if I trace, I don't know if you can see this well, if I trace with my finger around the actual outside of the mitochondrion, that's the outer mitochondrial membrane. If I trace with my finger around the inner membrane, you notice that's a lot longer tracing process because there's a lot more inner membrane than outer membrane. So inner membrane, outer membrane, there's a space between those two membranes, and then there's a space inside the inner membrane. So I'm going to go back a slide now. No, I don't need to go back a slide now. So notice this little box, they're blowing it up to a bigger picture. Here's the outer membrane of the mitochondrion. Here's the inner membrane. And here's the very inside space. This orange is the space between outer and inner membranes. And then we blow it up more to look at this picture. So here's the space most interior in the mitochondrion, and this is called the matrix. Mitochondrial matrix. It's the most inside. This is the inner membrane. This is the space between the two membranes, and in this picture the outer membrane is not shown. So the citric acid cycle stuff happened in the matrix. Citric acid cycle was happening inside the mitochondrial matrix. So let me go back and show that. Like here would be the matrix, so on. That's where the citric acid cycle is happening. So all of those products of the citric acid cycle are here in the matrix. And remember that some of those major products were NADH and FADH2 that were carrying energized electrons. They are going to pass their energized electrons to this series of proteins that are found in the inner mitochondrial membrane. So these purple blobs, they're proteins and they're enzymes, and they are members of the electron transport chain. So they are picking up energized electrons from NADH and from FADH2 and passing them one to another. And as they do that, the energy of the electrons goes down. The last member of the chain passes electrons to oxygen. That's electron transport chain. So to me, the cool part about the electron transport chain is that each one of these proteins in the chain has a preference for the energy of electrons that they pick up. So this, if I compare this to a game of hot potato, I hope you still play that. Like when you pass something from one friend to the next, to the next, to the next, and then when the music stops, whoever's stuck with it is the loser. So they're passing electrons one from the next, and the first proton in the chain prefers to get electrons that are very energetic, very high energy, to pick those up directly from NADH2 or from FADH2. The next protein in the chain prefers to pick up electrons that are slightly less energized. And the third protein in the chain prefers to pick up electrons that are even less energized. So in a way, this relates to how the electrons are going down in energy as they pass through this chain. But also, energy is not ever completely lost from a system. So where is their energy going? I'll come back to that in a minute. But so the first protein in the chain wants high energy electrons, the next medium energy electrons, the next low energy electrons. And then at the end, the oxygen picks them up when they are very, very worn out, super low energy electrons. Hold that thought just for a minute.